Thank you so much, Dr. Meekins, for the introduction. As Dr. Meekins said, I'm Sara Kearney. I'm a fifth-year graduate student in the Brennan Research Laboratory at the University of Florida. I'm really excited to be here with you on this platform to talk about my research, which involves designing and synthesizing axially chiral cannabinols to assist in the effort for developing new drugs in the cannabinoid sphere. So cannabinoid natural products are the cannabinoids that are found in the cannabis plant. So these are the chemical constituents of the plant that have all of the bioactivity associated with the use of this substance. And so many of these are pretty hot topics in the research sphere currently. And I'll just go through a couple of examples of these that you may be aware of. So the first of which is tetrahydrocannabinol, or THC. This is really the most commonly researched and commonly known cannabinoid in the plant. It's responsible for the wide variety of effects that are experienced with cannabis use. It's responsible for the majority of all the bioactivity that's observed. And these include properties such as analgesia, anti-emesis, anti-anxiety. And these are all really positive effects, therapeutic benefits that are imparted by THC. But unfortunately, there is the liability of its psychoactivity, which make it somewhat less ideal for a drug substance for therapeutic use. Cannabidiol is also very commonly known, becoming more and more popular lately. Its use as an anticonvulsant, anti-inflammatory, as well as a mild analgesic has been a really hot topic in the drug discovery field currently. And cannabidiol is considered non-psychoactive. So unlike THC, it can be utilized without the attendant liabilities of psychoactivity. And then finally, cannabinol, or CBN, is sort of a lesser well understood material, but it was actually the first molecule that was isolated from the plant. And so this is sort of the original cannabinoid natural product that was isolated back in the early 1940s. And it was found to have mild analgesic and antibacterial, as well as anti-anxiety properties. And it's considered very weakly psychoactive or non-psychoactive. And so unfortunately, it doesn't have really great therapeutic properties in terms of potency. So that really means in the drug discovery world that you need a lot of this material to impart any real therapeutic benefit. And so for that reason, it's been sort of sidelined as a lead for drug discovery. But we're definitely interested in this material in the Brennan Laboratory. So I'm excited to tell you more about why we are interested in this molecule. Just to give a brief introduction into the cannabinoid biosynthesis. So the plant can really efficiently manufacture these chemicals, starting from materials like this methyl allyl diphosphate and this isopentenyl diphosphate. These combine to form, with the loss of diphosphate, to form this diterpenoid fragment. Meanwhile, simultaneously, acetyl-CoA derivative and a malonyl-CoA derivative are combined to form a polyketide species, which then cyclizes and aromatizes to form a resorcinol type fragment. So this is an aromatic fragment and a diterpenoid fragment. For those of you who are not terribly familiar with the terms of organic chemistry, that probably doesn't mean a whole lot, but suffice it to say, the vast majority of cannabinoid biosynthesis is comprised of some sort of cyclic product that looks kind of like this, and some sort of acyclic product that looks like this, combining in various ways to generate this really key intermediate called cannabidiol. So cannabidiol is one of the main cannabinoid natural products found in the plant, and in the biosynthetic pathway, this is a key intermediate from which other cannabinoid natural products are formed. So specifically, once cannabidiol is formed in the plant, it can then be derivatized through a variety of different biosynthetic processes to the following cannabinoid natural products, including cannabichromene, cannabidiol, and tetrahydrocannabinol, so THC, that really popular molecule that is of great discussion for therapeutic development. So as chemists, we're really interested in mimicking the biosynthetic pathway. So we would really love to be able to do what nature does and make molecules as efficiently and as well as nature. And so to do that, we try to mimic the processes that the plant undergoes in its synthetic process to really efficiently generate molecules that are of interest for biological activity. And so in the laboratory, we can take molecules like this resorcinol, that's called Livitol, And we can couple it with a variety of these terpenoid fragments using a series of chemical reactions to target this intermediate. This is actually cannabidiol. So synthetically, it's most straightforward to take olivitol, couple it with a variety of terpenoid fragments targeting a cannabidiol intermediate, which then can be cyclized to form tetrahydrocannabinol. And from tetrahydrocannabinol, we can convert this molecule to cannabinol and other products as well. And so the essential thrust of this effort is, in the synthetic domain, is to take really simple starting materials, combine them in strategic ways to target key intermediates that can then be elaborated to the final target molecules. And to do this, again, as efficiently as nature, but in a laboratory setting. And the reason that we're interested in this is because cannabinoids are really interesting for therapeutic use, as I've mentioned. That's because they act on the endocannabinoid system. And this is a fairly newly identified biological system. It involves a complex system of G-coupled protein receptors that are involved in neuro and immunomodulation. So the cannabinoid receptors are primarily located in the central nervous system and the peripheral nervous system. And it's implicated in a variety of neurological disorders, as well as inflammation, epilepsy, and analgesia. So I will say in the analgesia domain, cannabinoids are of great interest for alternatives to highly addictive opioid substances. And so broadly, there is great utility for these kinds of molecules in a medicinal setting. And that's evidenced by 3 fairly new FDA-approved cannabinoid drugs. So Marinol, which is essentially just a THC-derived drug, was approved as an antiemetic as well as an appetite stimulant. Nabilone, this is a synthetic cannabinoid. So synthetic meaning this is not found naturally in cannabis, but has been synthesized and designed in a laboratory. And then most recently, Epidiolex was approved in 2018 to treat various forms of childhood epilepsy. And so these materials are really have a fairly, as I said, broad utility, but primarily our interest is pursuing them as potential alternatives for opioids, as the opioid epidemic has posed a great need for the development of new molecules that can treat acute and chronic pain without the liabilities of addiction. And so despite the fact that there are many cannabinoid natural products and they have great utility, there are still several very underexplored phytocannabinoids in the research sphere. And so those include cannabinol, cannabichromate, and cannabigerol. These are all molecules I showed on my first slide of the 5 main natural products in the cannabis plant. But in the Grenning lab, we are primarily interested in cannabinol specifically, or CBN. And the reason that we're interested in CBN is because just to compare the structure of THC, tetrahydrocannabinol, and CBN. CBN is essentially what we'd call an oxidation product of THC. So if you compare their structures, they're very similar. The primary difference being that this is a cyclohexene ring in THC, whereas we have a benzene ring in CBN. So this is fully oxidized. And actually, under ambient conditions, THC will oxidize to CBN at a fairly steady rate. And so this oxidation process is very favorable and is actually one of the reasons that THC as a drug substance is not ideal, because it is not terribly stable under just regular out on the counter conditions. But THC as a drug substance has some benefits in that it has a really high binding affinity for the cannabinoid receptors CB1 and CB2. So it has really good potency and efficacy. So when it binds and does what it does it very well. But the cons of this kind of molecule is that it's unselective. So it binds to multiple targets. It's strongly psychoactive, and it's poor stability. Again, that process of oxidation of THC being very favorable. So that's a lack of stability in that molecule. And that's something not very ideal for drugs, as you want the drug molecule to stay as it is intended. Comparatively, cannabinol, some of its benefits is it's very stable. So this, as I've highlighted here, this biaryl structure is very stable for drug molecules, very common in drug molecules, as it's easily functionalized to make derivatives. It's stability under various conditions. And then cannabinol as a product is not very psychoactive, or is considered very weakly psychoactive. So there's a bit of a debate in the literature about whether or not it's psychoactive, but the general consensus is that if it is, it's so weakly so that it's really irrelevant for a drug development perspective. But unfortunately, it is also fairly unselective, so it binds to multiple targets. It has a much lower binding affinity than THC and much lower potency. But the Grenning lab was inspired, though, by its structure, specifically by its biaryl core, because we saw an opportunity to impart a unique property of biaryls, which is known as axial chirality. And so axial chirality is when there is, so in this case, the axial chirality is affected by taking this methyl group at the C9 carbon and effectively transposing it to the C10 position. And what happens when you do this is now this methyl group and this hydroxyl group are close enough together that the molecule wants to twist out of two-dimensionality to allow for enough space between those groups so that you don't have electrons bumping into each other. And so that imparts a property of biaryls called axial chirality. So that means that the molecule has more three-dimensional shape, but that's due to the fact that about this bond here, these two aryl rings are now twisting out of the plane. And that's unique to biaryls and related molecules. And so in 2020, we reported my predecessor, Dr. Pramali Navaratne, who spearheaded this project. She published this really nice paper about the design and synthesis of this first generation axially chiral cannabinol molecule. So if you're interested, please check that out. It's really, really wonderful work and the foundation for my research project. And so axially chiral cannabinol, or axCBN as we have called it, the hypothesis was that a molecule like this would have the stability of cannabinol and the ease of functionalization of cannabinol, being a biaryl. So meaning it's easy to change out the groups at the various positions on the ring here. But it hopefully with greater three-dimensionality would have higher binding affinity for potential protein targets. And this is born of the fact that it is well known that biological targets have a very specific three-dimensional shape. So ligands or molecules that can bind to it that have a very specific three-dimensional shape are likely to be stronger at binding and more selective for those targets. So three-dimensionality is an important component of drug discovery. So hopefully with that, if it has enhanced binding affinity, it would have a greater potency and efficacy. And then hopefully it would also retain the lack of psychoactivity that is known with the cannabinol natural product. So the hypothesis was that axCBNs would be three-dimensional, and this was confirmed via computational work that was done by Dr. Adrian Reutberg in his lab at UF. So what they did was they modeled a THC-like framework. And so they were looking at the angle around this series of bonds here. And so they determined that it's called a torsional angle. And this torsional angle is basically a measure of how three-dimensional the molecule is. And so we also in synthetic organic chemistry like to designate three-dimensionality by these wedged and dashed bonds. So when you see those, that means that the molecule is three-dimensional about those points. And so for THC, the torsional angle is about 48 degrees. And if you compare that to cannabinol, the torsional angle is only about 19. So THC is significantly more three-dimensional than CBN, but it was computed that if you affect this alkyl transposition, meaning you take this C9 methyl group and you transpose it to the C10 position, forming axially chiral cannabinol or axCBN, it was computed that the torsional angle in this molecule would now be 38 degrees. So that is far more similar to that of THC than the natural product cannabinol. So this sort of confirmed that axCBNs would have greater three-dimensionality due to axial chirality. And so just to show how we like to represent this, when we use these bold bonds here, that's how we're showing that these regions of the molecule in bold here, you can imagine those sort of coming out at you, so coming out of the plane. And so to really see what that would look like, we can also sort of draw it in this really suggestive way where you can kind of see this aryl ring is really sort of twisted out of the 2D space. So hopefully that's not too confusing. We try to represent them like this as much as possible, just to really help drive home the point that these molecules are not flat, they're not planar, they have significant three-dimensionality. And so a property of these kinds of axially chiral molecules is called atropisomerism. So atropisomers are molecules that display axial chirality, and they equilibrate between two different conformations that are known as enantiomers or atropisomers. So if we consider the planar rendering of these molecules, so we can draw these molecules very flat with no three-dimensionality indicated, but really in their ground state, meaning in the lowest level of energy they exist in, they're going to be preferentially three-dimensional. And that's because, as I had explained earlier, when you have substituents very close to this axis of rotation, these are essentially bumping into each other, and to minimize electronic repulsions, the molecule is going to twist out of to minimize those interactions. But it can twist in two different ways. So it can twist such that you have these axCBNs enantiomer, which is just one conformer, but it can then pass through this planar intermediate and settle back down into this other enantiomer, the P enantiomer, wherein it is twisted, but twisted in the opposite direction. And so this is a key property of atropisomers. So at room temperature, depending on the energy barrier associated with this process, and that's what this coordinate shows. So this on this axis here is delta G is representative of the energy associated with the process. And so if you can imagine starting from this from this enantiomer, we must climb some sort of energy barrier to pass through this higher energy planar intermediate, and then it can relax back down into the opposite enantiomer or atropisomer, wherein it's twisted in the opposite direction. So that's a key characteristic of these molecules that I'll discuss later on. So to really showcase the possibility of these molecules, Dr. Pramali Navaratne in our lab, she developed a really concise, beautiful synthesis, first generation synthesis of this molecule. And so I'll just summarize it here. So in 4 steps from olivitol, so this is olivitol, this is a commercially available starting material. In 4 steps, she was able to build this aeropropargyl ether intermediate that she could then perform an aldol condensation using allyl cyanide to form this diene species. And this diene can then, under thermal conditions, undergo a 4+2 cycloaddition. So essentially forming bonds between this alkyne moiety and the diene species, cyclizing to this cyclohexadiene. And I will note that unfortunately in this step, so taking this allyl cyanide and condensing it in with this carbonyl species, I've drawn this as a squiggly line to indicate that this, that the geometry about this bond can either be such that you have this really nice diene wherein you have both double bonds poised to undergo reaction with this molecule, or you can have the situation in which the position of these two groups is flipped. And in that case, it's not designed to undergo that desired cycloaddition. And so instead, what you obtain is this coumarin product, or sorry, this benzochromine product, resulting from a different kind of chemical reaction. And so this is sort of one of the liabilities that happens with chemical synthesis, is you set out to synthesize a particular target molecule as drawn here, but there are multiple outcomes in your reaction. And so one thing that can happen is that you end up with a mixture of double bond isomers, in that you have what is called the Z isomer, which undergoes the desired chemistry, or the E isomer, which is not structurally able to do so and instead undergoes a different reaction, yielding an entirely different product. So that's just something that happens quite often in the laboratory. And so we have to deal with those unwanted outcomes strategically. But unfortunately, we were able to obtain enough of this desired cyclohexadiene product, and through oxidation using DDQ, we could obtain this biaryl species that was then in 4 steps elaborated to the final target molecule of axCBN. And so this synthetic protocol, I did abbreviate it here, but if you're interested in seeing the full synthetic route, again, please check out this paper in ChemMedChem. But this synthesis of axCBN was performable on the gram scale, so that allowed for preliminary biological investigation that I won't talk about in this presentation, but if you're interested, please absolutely put some questions in the chat. I'd be happy to address those. And so we were really interested, I'll just toggle back here for a second. So we were really interested in continuing to explore axCBN chemical space, and there's a variety of reasons for that. One is the really encouraging preliminary biological data that's reported in this paper, suggesting that these kinds of molecules are active. They are less psychoactive than THC, so we really wanted to continue to explore those. But the synthetic protocol, unfortunately, did not allow access to quite the variety of new chemicals that we wanted. So, for example, in the axially chiral cannabinol structure, there is now no methyl group at the C9 position. So in the natural product, there is a methyl group just like this one right here. And so to really fully understand the importance of a methyl group at the C9 position, we need to be able to reinstall it in the axCBN framework. And unfortunately, this synthetic approach did not afford the ability to do that. And so we would like to be able to synthesize molecules with functional groups at this position, as well as address a couple of the liabilities associated with this first generation route. And so where I came in really for this project was trying to spearhead a new reaction development, new synthetic development to target further analogs of axCBN. And so the first analog that we were really interested in targeting is this sort of 9-methyl axCBN, where, as I said, this methyl group that had been moved from C9 to C10 has been restored. And so we imagine this coming in 3 steps from this really key biaryl lactone species that could be derived in 2 steps from this coumarin diene. And the logic behind targeting this coumarin diene species was really that in its synthesis, we could control the orientation of the bonds surrounding this olefin. And that was important, as I said in the previous synthesis, we would get a mixture that only part of the mixture would undergo desired chemistry and the other part was sort of sacrificed. And so this would be a way to fully control that reaction and obtain this material in higher yield. So to do that, we started off with a series of these aldehyde resorcinol derivatives. And through very established literature protocols, we can take dimethyl propargyl chlorides and acrylic acids and couple them in strategic ways to generate these alpha beta unsaturated esters that in the presence of a base like DBU can undergo a condensation reaction to form a bond here with the loss of water, forming this coumarin diene. And so this was a key intermediate in our synthetic protocol. And that's because we hoped that if we could achieve these kinds of molecules in good yield, then once we subjected them to heat, they would undergo a 4+2 cycloaddition involving this region of the molecule, furnishing this cyclohexadiene species that could then, through subsequent transformations, be elaborated to our target molecule. But unfortunately, once we had this coumarin diene in hand, what we observed instead was not a 4+2 cycloaddition, but instead something called a 3,3 sigma tropic rearrangement. And so that's essentially where chemistry is not occurring between these regions of the molecule, but instead is occurring between this alkyne and this position on the aryl ring, forming this highly conjugated, very stable pyranocoumarin type species. This is not entirely surprising. The 4+2 cycloaddition that we wanted to observe is fairly kinetically challenging, meaning that it has a high barrier to activation. So all reactions have to pass through some energy barrier in order to achieve the transformation from starting material to product. And depending on how high that barrier is, that sort of determines in part how difficult that transformation is. And so in our case, we're really asking a lot of this molecule. So I've tried to demonstrate through coloring here. This is a fully aromatic, fully conjugated system. So all these double bonds are in conjugation with one another. And that exists in 2 different resonance forms, meaning you can move some electrons around and show in multiple structures that this is a very, very well conjugated, fully aromatic molecule. However, upon 4+2 cycloaddition, you can see here just by the decrease in this blue coloring here, we've really broken some of that conjugation and aromaticity. So we take a fully aromatic molecule and we make it partially dearomatized. And so that's a very high energy cost as this is a highly stable species. So we're asking it to become less stable as it undergoes the transformation. And so we're asking essentially for it to go from fully conjugated to partially conjugated, fully aromatic to partially dearomatized. Whereas in the first, or I'll just show sort of mechanistically why that happens, but just to say this penalty is not a problem in the first generation synthesis. This is something we didn't see before in the first route. And so this is very common. You try to solve one problem and then you end up creating another problem. But, I will disclose that we did find a way around this. But the reason why, in addition to the fact that this sequence is very unfavorable energetically, the alternative that actually does take place is exceedingly favorable. And that's because when you have a flow of electrons producing this allynyl intermediate that can then tautomerize to the corresponding phenol, a 1,5 hydride shift yields this 6-pi electrocyclization precursor that, upon cyclization, generates this incredibly thermodynamically stable molecule. So we call this a thermodynamic sink, meaning that this is about as low in energy as you can get in terms of a structure like this, starting from this kind of material. Ending in a structure like this is about the most thermodynamically stable and favorable process you can undergo. So it's really hard to fight against such a favorable series of reactions. So on two fronts, we're struggling here to get our molecule to do what we want it to do. And this is a really cool introduction, I think, into the concept of Curtin-Hammett control, which is a physical organic chemistry concept. So essentially, this principle states that when you have molecules that can interconvert between two different conformations, such as this material here, where we can draw this propargyl moiety sort of poised up toward the diene component, or we can draw it, imagine it swinging down and located more proximal to the arene component. This process of that propargyl arm sort of swinging up or down is happening very quickly at room temperature, and certainly at higher temperature conditions. So when we have an equilibration situation like this, and wherein each conformation undergoes a distinct reaction pathway in that, so if we imagine aryl propargyl ether arm up, poised to undergo 4+2 cycloaddition to yield the cyclohexadiene species, versus the arm swinging down for 3,3 sigma tropic rearrangement, yielding this pyranocoumarin. So there are two different pathways, and they're both require distinct inputs of energy. The difference in the product distribution, so if you imagine both products are possible, you could potentially get a mixture. The distribution of products is going to depend on how different the barrier to each pathway is. And so I've tried to represent that on an energy diagram here. So if we imagine conformer A and conformer B, some low barrier to interconversion between the two of them, and each respective pathway having its own energy barrier that it has to cross. The Curtin-Hammett principle tells us that the distribution of products is going to depend on how great the difference in those two barriers is. And so the fact that we routinely we're seeing exclusively product D, that informs us that this barrier difference is so significant that we're seeing exclusively one product over the other. And so there's various ways that we can think about promoting the desired pathway synthetically, and those include solvent effects. So changing out which solvent you're using can stabilize transition states. So transition states are this sort of high energy intermediate. As A moves to C, it's going to pass through some high energy intermediate. Lactone reduction, meaning, if we took this coumarin diene species and we broke this lactone bond, so doing that preemptively would break the aromaticity of this coumarin species. So we would prepay that loss of aromaticity penalty before cycloaddition. And then substrate engineering. So some ideas we had thought about in this domain were to increase the steric bulk of this substitution here. So if you imagine making this really bulky, perhaps then cyclization at this position, if it was crowded by this R group, maybe that would be less favorable. But unfortunately, each of these attempts to convince our molecule to do what we wanted to do was unsuccessful. And so finally, to make a long story short, we turned our attention to metal catalysis, which is what I'll tell you briefly about right now. So metal catalysis introduced us to the world of catalytic cycloisomerization. So it's known in the literature that you can use low valent metals to catalyze oxidative cyclization processes that can net 4+2 cycloadducts. So taking our coumarin diene species in the presence of rhodium-1, it can undergo a cyclization to form this rhodium metallocycle that can then further coordinate the diene component. And then this really sort of templates the molecule for an alkyne insertion, yielding this species here. And then upon a reductive elimination of that metal, we can unveil the desired cyclohexadiene. So this is fairly precedented, but the key component here was that we chose rhodium as a metal. People often say, why did you choose rhodium? It's so expensive. And it's a very good question. But the rationale was that rhodium-1 is known to act mechanistically in this way. And so other metal species you could think of like copper or gold, while they would template the molecule for cycloaddition, they would also potentially and likely activate, really exclusively activate the alkyne component of the molecule. And so the fear was if we activate this component, that would also probably lower the barrier to the cyclization happening between these two atoms. And so we really wanted to go with a metal that would exclusively activate the diene component, as that is the only part of the molecule involved exclusively in the desired chemistry and not the unwanted chemistry. And so to make another long story short, we did extensive optimization of various rhodium catalysts and silver triflate additives. and solvents and temperatures, and landed on these conditions as the optimal reaction conditions. So taking this coumarin diene starting material in the presence of 5 mol% of rhodium cod dimer with a silver triflate additive and trifluoroethanol at room temperature, we could achieve 100% conversion of our starting material to both a mixture of the wanted cyclohexadiene and the actually in situ oxidized product. So essentially what happens here is upon cycloization, it's very favorable for this molecule to oxidize to the corresponding biaryl. So we were seeing that happen without addition of any external oxidants. So getting a mixture of these two molecules was very favorable in that the next step with this molecule would be actually to convert it to this molecule so that this happens sort of on its own is exciting. And really the most important thing about this result is that absolutely none of this pyronocoumarin product was obtained. So we were able to tune the reaction trajectory to exclusively produce a mixture of desirable products and totally shut down that undesired reactivity. And so, yeah, as I said, the next step would be to take the cyclohexadiene and oxidize it. So any unoxidized cyclohexadiene, we're able to oxidize with DDQ to the corresponding biaryl and good yield. So a really successful series of reactions. And this new reaction gave us access to really key intermediates for our synthesis of new axially chiral cannabinols, which is really the main goal of this research program. So we were able to take a variety of starting materials and synthesize a diversity of these coumarin dienes, wherein we can vary the substituents at this R1 position, the R2 and R3 position, as well as R4 and R5. You can imagine various building blocks coming together strategically to construct these kinds of scaffolds. And then with our newly developed rhodium catalyzed conditions and oxidation protocol, we can obtain a variety of biaryl lactones that can be further elaborated to axially chiral cannabinols. And so we explored the scope of this reaction, but the few that I really want to focus on this evening are the ones that are most relevant to axCBN drug discovery. And that is the sort of model substrate, as we call it, where there is no substituent except for a hydrogen at this position. The N-pentyl chain, which is the alkyl substituent that's part of the natural product cannabinol. And then this dimethyl heptyl chain. And this is a very common substituent that is found in cannabinoid literature. It's known to impart enhanced potency and efficacy. So it was sort of a natural analog for us to synthesize for our axCBN series. So what I really, just to summarize what I've told you about thus far, is how we developed a catalytic approach to overcoming undesired innate reactivity. So we had this molecule that can equilibrate between two different conformations and innately wanted to undergo an undesired reaction pathway due to the fact that this is a lower energy barrier compared to the chemistry we had been looking for. And so in order to lower that barrier to activation, we utilized rhodium catalysis, which effectively lowers the activation barrier so that now this is the favorable pathway. So you can see here, lowering the activation barrier so that now this is the higher energy pathway so that we could selectively obtain this cyclohexadiene. And so that was sort of an unforeseen new reaction development component of my research, as we started out with a very target-oriented project, but found out that we would need to develop a new method to affect the overall transformations that we wanted. And that's very common in synthetic chemistry, but it was exciting to be able to leverage the power of catalysis to tune our reaction trajectories in an effective way. But this is really, again, a target-oriented project. So we took our biaryl lactones that we were able to achieve through rhodium catalysis and continued on in our synthetic approach. So the next step in this protocol is to use a reductive agent, lithium aluminum hydride, to break this lactone bond to unveil the corresponding diol species. And then these diols can be acylated using acetic anhydride and pyridine to form the corresponding bis acetates. And so this series of 6 molecules were our most interesting molecules to date, in that they all exhibit axial chirality, but will have different barriers to interconversion between those atropisomers, as I've explained, given the fact that there's a significant difference between hydroxyl groups versus acetate groups. And so the reason that we synthesized both series was strategically to sort of probe the effect of structural modifications on the barriers to equilibration of the atropisomers. And so we synthesized small collection of axially chiral cannabinols, both for study of their atropisomerism properties as well as their biological activity. So again, that atropisomerism, meaning these molecules can then exist in one conformation. They pass through some planar intermediate, and this has some sort of energy associated with it, and then they relax back down into the other enantiomer. So we wanted to study what the energy of that process was, and that can be done in a series of very cool experiments that I'd be happy to share more about if people have questions. But suffice it to say, we performed those experiments thus far with these two molecules. So sticking with the N-pentyl substituent, we compared the barrier to atropisomerism for the diol and the bisacetate. What we found is the energy barrier associated between the equilibration of each enantiomer for the diol species was about 13.7 kcals per mole. So for reference, at room temperature, that there's about 20 kcals per mole worth of energy. So the fact that that's lower than 20 this is lower than 20 kcals means effectively that at room temperature, this atropisomerism process is happening very readily, very quickly. These molecules are interconverting rapidly between the two different conformations. And what that tells us is that under biological conditions where the temperature is 37 degrees Celsius, that would be higher energy available. So that means the molecules are going to be interconverting at a greater rate. That there's certainly access to both enantiomers at the biological target. So essentially the biological target has access to this enantiomer and this enantiomer, and it can discriminate which it prefers, which has stronger binding. And so for early drug discovery, this is actually kind of advantageous. We don't know exactly which conformation is preferable. So the fact that it has access to both can help us understand which conformation is better at binding. Conversely, the bisacetate molecule has the barrier to atropisomerism of about 17. So a difference in 4 kcals per mole is very significant. So that tells us that this equilibration process is happening much slower compared to this one, though it's still under that 20 kcal per temperature sort of threshold. So it's definitely still happening pretty readily at room temperature. But what that tells us is that as you start to increase the steric, as predicted, as you start to increase the size of the substituents at these positions, you're going to slow down that interconversion because those groups are really bumping into each other in space, and so they're not able to move quite as rapidly. And so this would tell us that if we wanted to strategically synthesize a series of molecules where now instead of being able to interconvert, they're locked in one conformation or the other, we could start to get a sense of what kinds of substitutions we would need to put there to affect that change. So very excitingly, we've had some preliminary biological data developed by a collaboration at the Research Triangle Institute in North Carolina. So Dr. Tom Gamage and his team have very graciously performed preliminary binding affinity assays with the 6 molecules I've reported here. And so the 5 molecules of interest from that screen are shown here. And just notably, the cannabinol natural product was used as a control, as well as the CP55940 molecule. This is a synthetic cannabinoid that has high potency and efficacy and is used very commonly in drug discovery research. This was actually made by Pfizer, but never released for actual drug product, but it's just used strictly in research. But it's a very potent activated molecule toward the CB1 and CB2 receptors, the cannabinoid 1 and cannabinoid 2 receptors. And so these are really good molecules for sort of gauging where our molecules fall on the spectrum of sort of fairly inactive. Cannabinol is considered fairly inactive, whereas the CP55940 is pretty active. So we can kind of see where our molecules fall on that spectrum. And so axCBN1, this is the first generation axCBN diol, synthesized by Dr. Navaratney. You can see this has a 1.3 micromolar to greater than 3 micromolar preference, affinity for CB1 and CB2. So these, just to briefly introduce these receptors. So these are the canonical cannabinoid receptors found in the human body. CB1 is primarily located in the central nervous system. CB2 is primarily located in the periphery. And so CB1, unfortunately, most modulation of CB1 is related to the psychoactive properties of THC, whereas CB2 activation seems to be fairly innocuous in the psychoactivity domain. And so in cannabinoid research, we're really curious to see if we can find molecules that are selective for either CB1 or CB2. Typically CB2, as I said, is not associated with psychoactivity. But in this early stage, we're really just curious how these molecules are binding to the receptors at all. And so the first generation axCBN1, we're at micromolar affinity. That's not great, but still encouraging to know that this is a synthetic molecule and it is in fact binding to the desired targets. axCBN2, which is a second generation molecule, has improved activity. So we're now below 1 micromolar for each of CB1 and CB2. But then the most exciting molecule thus far is this axCBN3. So now we have swapped out this N-pentyl chain for that really active dimethylheptyl chain. And now we are below 1 nanomolar binding affinity at CB1 and below 1 nanomolar at CB2. So this is even more potent than the most potent synthetic cannabinoid that's really out there for in the research field, the CP55940. So we're very excited about this and definitely going to be following up on this molecule with additional experiments. And then the other very interesting result to us is that if you take these diols and you acylate them to yield axCBN5, you go from sub-nanomolar activity to now what you have is greater than 1 micromolar activity at CB1, but very low micromolar at CB2. And so what's interesting about this to us is that now you have a 6-fold preference or selectivity at CB1 over CB2. And so in drug discovery for cannabinoids, finding a selective CB2 modulator would be very exciting, because if we could selectively target CB2, that opens up a lot of possibilities for imparting the desirable cannabinoid effects without the attendant psychoactivity liabilities. So this is really exciting data for us. And so at this point, we're really interested in continuing to develop new molecules to kind of probe further what kind of structural modifications to these scaffolds will impart more beneficial properties. So, how can we change functional groups at these positions, at this position, at this position, basically sort of renovating the molecular structure to see how we can improve the biological activity further, knowing that these molecules are very active. So in summary, hopefully I've introduced to you guys that we have a new platform for drug discovery inspired by cannabinoids. And that's really made possible by axial chirality. So capitalizing on axial chirality can really open up new directions for drug development in this space. So taking this very flat cannabinol molecule, imparting 3-dimensionality through axial chirality to make it a better drug molecule can open up a really, really fun new direction for drug development. And that we have very encouraging initial preliminary data suggesting that these kinds of molecules are in fact active and do bind with significant affinity for the cannabinoid receptors. And so at this point, we're trying to develop new ideas for how we can expand this platform and new chemistry, as well as new molecules to synthesize. And this work is going to be coming soon to a journal near you. So if you're interested, please be on the lookout for that. Hopefully in the next month or so, we'll hopefully getting that submitted. And so with that, I would just like to say thank you very much for your attention. And a big thank you to my PI, Professor Alex Grenning, for giving me the opportunity to work on this project and for his mentorship and support. And all the Grenning Lab members, past and present, who have just supported me and been a wonderful group of people to work with. I really can't say enough about the team. This is the sort of the original team I joined, and then all those wonderful senior students graduated, and here's where we are now, and even several of these people have left, but it's really still just a fantastic group of people to work with, and I'm so privileged to be part of the team. Dr. Jan Gavirga for his instrumental analysis support, as well as Dr. Tom Gamage for his really exciting biological data generation that he's provided and biological screening. and Dr. Ben Meekins for this opportunity to speak with you. Our collaborators are Corteva AgriScience, who I actually didn't get to talk about tonight, but they're a great collaboration. RTI International, thanks to the University of Florida and NIH for funding. We are the Growing Research Laboratory, so please check us out. And if you're interested in following my research, you can follow me on Twitter or my website here, where I like to blog about chemistry. And so thank you again for all of your attention, and I would be more than happy to take questions. Yeah, thanks so much.
Fantastic. So that was a great talk. That very interesting work that you're doing. So we'll just jump straight into the questions. And just for the audience who they did turn out for you, they're more than welcome to continue asking questions sort of as we go. So we'll just go through them until there are no more questions. So let's get started. So the first question is on slide 11, where you're talking about the torsional angle. Could you further increase that torsional angle, and would you want to?
Yeah, absolutely. So that's actually the main idea behind designing new axially chiral molecules. So the degree of three-dimensionality is determined by really how much the substituents at these two ortho positions about this axis, how much they're bumping into each other. And so the degree to which this bond, there's going to be rotation about this bond, meaning that twisting out of the 2D plane, that degree of twisting, which affects the torsional angle, is going to be entirely dependent on the steric or bumping into each other that is happening between these two species. So one reason we're interested in quantifying the energy barrier between twisting between different conformations of the molecule is to kind of give us an idea of, okay, how much steric interaction is happening here, such that that twisting is hindered. And the more hindered that it is, likely the greater extent that it will be twisted in one conformation or the other. And so that really helps us design new molecules by understanding, well, when we make structural modifications at these positions, what does that do to the torsional angle? And so we'll continue to explore that as we design and synthesize new axCBNs.
So this is a question I had for a non-synthetic chemist. So this is on slide 13. How do you actually isolate the desired product?
Sure. So meaning the final.
The one where you had the two, I forget, I don't know if this is the right term or not, the two isomers. So the Z and E.
These two.
Yeah. Mm-hmm.
Yeah. So unfortunately, part of the problem with this is that you actually cannot separate the two isomers.
Okay.
So in some cases they are separable. They might be different enough that you can separate them. In this case, unfortunately, they're not different enough. So what happens is they're basically, from separations techniques standpoint, they're virtually inseparable. And so unfortunately, you have to commit the entire mixture of these to the next step, this heating in toluene at 110 degrees. Mm-hmm. And that's why you're limited here, because it doesn't really matter either way, whether you have it as a mixture or not, you still can only isolate a smaller percentage of your desired product if a mixture is formed anyway. But yeah, unfortunately but that said, fortunately, once you do heat them, they do separate into these products that are very separable. Once yeah. So at that point they become separable. So you can isolate this material, and those techniques involve, it's called chromatography, and it's an entire other subject of its own, but it's very interesting if you're interested in further reading. But chromatographically you can separate these molecules, and then you can take this and just oxidize it, and then this can be set aside for whatever. Unfortunately, we haven't found great use for this molecule.
Just hanging on to it just in case. Okay.
Just in case.
Yeah. All right. Let's see. So on slide 21, does the rhodium catalysis lend itself to larger scale synthesis, or is one of the next steps to figure out an alternative route that doesn't have to use rhodium?
Yeah, definitely. Yes, we definitely would like to find a different route. So unfortunately, rhodium was wonderful in that it provided exactly what we needed, especially on a small scale, was that we could tune the reaction trajectory as we wanted. And that was a really exciting result after many, many months of only ever seeing this product.
Sure.
Everything I tried again and again. So yeah, unfortunately, rhodium is not, certainly no industry lab would touch this. They're not going to say, yay, we have a rhodium catalysis method for synthesizing large quantities of axCBN. So unfortunately, no. And we definitely are thinking about ways to overcome the selectivity problem, namely by modifying the substituents here. And right now that's mostly involving, can we take these two methyl groups and bind them up in a small ring so that, and I can just toggle back quickly to the mechanism here. So if these groups are not free and they're bound up, say, in a 4-membered cyclobutane ring, what will happen when you move from here to here is that now this being bound up in a ring with this external double bond, that's a ton of ring strain. And that's really unfavorable from a structural energy standpoint. So we're hoping that we can shut down this process if we can induce that strain element, that perhaps this will become so unfavorable that now this process becomes the preferred mode of reactivity.
Interesting. Okay. Let's see. So on slide 24, is the goal for binding to the receptors to just, are you trying to beat out the natural products in the known synthetic compound, or is there some other target you're aiming for in terms of the binding?
So we'd certainly want to exceed the binding profile of CBN. So when you're looking for potency and efficacy in drugs, and this is a little bit different. We're not really studying potency and efficacy at this point. It's mostly just how strongly does it bind to the target. But usually binding affinity correlates directly to potent efficacy. Because if it binds more strongly, it's more potent, more efficacious. You need less of it to do what it does, and it's more efficacious. So we'd certainly want to exceed the affinity of CBN, the natural product. So really, we're looking for less than we're looking for a nanomolar to under nanomolar scale for these kinds of molecules. It's for central nervous system drugs of any kind, the lower dose, the better. So we would love to see the lowest. I mean, and generally with most drugs, you want to see lower doses, but especially with central nervous system drugs, the minimum would be great. And then, so this is really the very, very first step, was just to see, does it bind? If it does, how well? And the next step would be further downstream assays that I'm not as familiar with, and I look forward to learning more about those in our collaboration with Research Triangle Institute. They'll kind of advise what the next steps are for these two molecules here that are the most promising.
Interesting. Okay. And you said in addition, so you want to find a stronger binding affinity for your molecules, but then you also want to try, ideally you would try and find one that has a preferential binding for either the CB1 or CB2. Is that right?
Yeah. So especially CB2. So unfortunately, to date, to my knowledge, there have been no selective CB1 agonists or antagonists. So they either amplify or diminish the natural response of the receptor, activity of the receptor. No molecules that selectively target CB1 that don't also impart psychoactive properties that are associated with THC intoxication. And so it would be great if we found a molecule, because CB1 has so many wonderful properties for therapeutic use, if we could separate that from the intoxication. So if we could find that, that'd be great. But I think the current state of the art suggests that it's really going to have to be a CB2 selective molecule that is able to be therapeutic, just because CB2 activation is not associated with intoxication at all. So that's sort of the logic behind that.
Okay, no, that makes a lot of sense. And then on this same slide, are the stereochemistries that you're showing here what are predicted, or are these based on NMR data or crystal data?
That's a really great question. I am just showing one atropisomer, one enantiomer. All of these molecules are what are called class 1 atropisomers. So that means they have barriers low enough to that equilibration between enantiomers. They have barriers low enough that they are fully equilibrating at room temperature and under also likely under ambient, which means thus under biological conditions, they are going to be fully racemizing. So I'm choosing to represent them as one enantiomer, but it's fully racemic, meaning there's both enantiomers in a 50-50 mixture at all times present. So yeah, unfortunately, I have yet to synthesize a molecule that is conformationally stable. And I would know that I like the synthesis, unfortunately, is also racemic. So even if I was to synthesize a molecule that was conformationally stable, it would be still a 50-50 mixture of each enantiomer, but I would then have a hope of being able to, separate them via certain separation methodologies. But right now I couldn't do that because they're always converting between the two, if that makes sense.
I got you. Okay. Let's see. Is there an enantiomer that could be more potent in one form versus the other?
Oh, absolutely. So really interesting examples of this I can think of just to exemplify how important enantiomers are is there's a drug called thalidomide, and that was developed in the early 20th century to treat nausea in early pregnancy. And so unfortunately, we didn't know about enantiomers and how important they were in drug development. And it turns out that one enantiomer of thalidomide helps nausea, and the other enantiomer is toxic to the developing fetus. And so that's to me a really, really good example of why conformation is very important in organic molecules. And so it could absolutely be possible. Another good example is cisplatin, which is an anti-cancer drug. Cisplatin is one enantiomer, very therapeutic. Transplatin, the other enantiomer, very toxic. So another good illustration there. So we don't know, and we would love to discern that through further synthetic efforts and biological assessment.
Okay, that makes sense. Let's see. Is the racemization a concern for drug discovery?
Definitely. Yeah. So, well, yes, definitely in some cases. So in atropisomerism, there are 3 classes of atropisomers. In class 1, they're fully equilibrating between enantiomers. Class 2, and I'll just say class 3, they are completely conformationally stable. And then class 2 is kind of in that intermediate stability where they are interconverting, but it's not rapid. It could be on the order of hours, days, months, years. And so the class 2 is really the risk in drug discovery, because those are if you can't you either want no control or total control. So in terms of conformation. Yeah. So if you have intermediate control, that can be a problem because you can't have real confidence that, for the long haul, your molecule's staying in one conformation or the other. And if you have that unfortunate case where one is, if it's just a matter of one's active, one's not, it's like, well, it just means your drug is becoming inactive over time. You could have the unfortunate incidents where one is active, one is toxic, therapeutic, and one is toxic. And that's where it becomes a significant risk. Gotcha. When you have those class 2.
So if you do have a case like that where you have that class 2 where, like you said, it's kind of the time period for them to change is long, are there ways, and let's assume some miracle drug, like, it does exactly what you want it to do, but only in the one enantiomer. Are there ways to try and push it to one side or the other without I guess, destroying, the target molecule?
That's a really good question. Yeah. So I don't know, firstly, how likely that would be, only because usually if it is a really active molecule, it's really active in its one conformation. So if during testing it's already converting to the other conformer, I don't know, like, depending on the time frame for the experiment, you might not see it as active because it is interconverting. If the time frame of the experiment is short enough that you don't see equilibration to that inactive conformer, I think at that point, likely the first thing that we would try would be, let's see if we can switch out those functional groups and just see if we can make them very similar, but just a little bulkier so it doesn't racemize. And that's hard because that's called bioisosteric replacement, where basically, you're taking one functional group and you're swapping it out for something that's so very similar that hopefully the activity is maintained. But it would be quite unfortunate if you have one that really needs to be exactly as it is. But I think that this is where this kind of gets out of my area of expertise for sure. But I think that's where you would get into sort of, what can formulation scientists do in terms of like, can we make this so rapidly releasing, so rapidly active that, it's getting into the body quickly, being absorbed, getting to its target, its active site and acting before it's degrading. Maybe it'd have to be something where it's just fast acting enough that it can do what it needs to do before it converts to that inactive species. But I don't know how feasible that is either. So this is definitely a challenge in an axial chirality drug discovery for sure.
Okay. Let's see. In your cyclization investigation, you mentioned a lot of factors towards helping the cyclization you'd want. You mentioned solvent effects. Have you considered solventless chemistry? If not, that's okay. That's our resident mechanochemist.
Is that solvent? Did you say solventless?
Solventless. Like neat? Yep.
Neat. Okay.
Yeah, he's a mechanochemist. Yeah.
Yeah. So we call that neat. Yeah. Unfortunately, the temperatures at which these kinds of cyclizations, they're called diels-alder cycloadditions, they need to happen at pretty elevated temperatures usually, unless they're catalytic, like I demonstrated, or really super favorable. And in our case, ours is not that favorable. So the temperatures that we need to do the reaction at, if you were to heat those materials neat, they would just degrade. They would not. What really needs to happen is they need to be dissolved in some solvent media to kind of, I kind of imagine it, and I don't know if this is technically true, but in my mind, I sort of imagine it as like, it's sort of insulating the molecule to all the heat, it's helping to absorb that heat energy. And so it's not just the molecule getting blasted with all of that thermal energy. So I think that's part of it. And also solvent effects, I was so hoping in the beginning that this was just a matter of finding the right solvent. There's a lot of literature that diels-alder reactions can be accelerated or promoted with just the right kind of solvent. And so I was so optimistic that would do it. But it's unfortunately, I think what happened is any solvent that promoted the diels-alder also promoted the other reaction that I was trying to shut down. So yeah, I just think that's where we're at. But yeah, solventless. I've only done in my entire 6 years of synthetic chemistry, I think I've done neat reactions. There's 2 reactions that I've done that are neat.
Okay. Fair enough. Yeah, that's Isaiah Spate. He's a mechanochemist, so he likes to beat things up with the balls and then get products out of it that way.
And it would be so much greener, right?
It
we didn't have to use solvent. So if we could do it, we would for sure.
Okay, there you go. See, there you go, Isaiah. You got it. Fantastic. It looks like that's all the questions we've got. So I'll give any last second ones that want to come in. Please feel free to do so. But I'll start wrapping up a little bit. Thank you to everybody for coming out. Thank you, Sara, for a fantastic talk. Really informative, very well done. So thank you for that. Like I said, as a chemical engineer, I came in knowing almost nothing. And now I understand it at least a little bit better. I probably won't be doing synthetic chemistry anytime soon, but that's okay. So yeah, again, thank you to everybody for coming out. If you are, I'm guessing that there's at least a few of your lab mates that are there that are watching this. So if any of you would like to give a talk as well, hey, reach out. I'm on Twitter at Meekins Lab, MEEKINS Lab. We would, I'd be happy to get you on here. You can show off some of your work too. And yeah, I think the last thing I'll say is I hope at least some of you will come back out next Friday, July 15th. We're actually doing an all-day conference live stream, calling it Science Palooza 2022, because I'm really creative with names. It will be going on starting at 9 a.m. Eastern Standard Time. Right now, we have 11 different talks from 6 different countries, so researchers from all over the world. Again, wide variety of fields. I hope you'll come out for that. Check out some of the work that's being done. And I think that's it. So again, thank you to Sara for a fantastic talk. If I can get you to hang around for just 1 minute, we'll talk very briefly. And for everybody else, I hope you have a great weekend. And like I said, hopefully we'll see you back here next week at 9 a.m. instead of 10:30 p.m. Yeah. Thanks everybody for coming out. I'll see you next time.